Hello everyone, this is Medical Gateway and today we're going to study Amoeboid movement and ciliary movement. So what exactly is Amoeboid movement and why Amoeboid movement is known as Amoeboid movement? Because it looks like amoeba, the movement of amoeba. Okay, so now let's discuss it. So this is a WBC which is going to form amoeboid movement. Along with the WBCs, fibroblasts and germinal skin cells also perform amoeboid movement. So what is happening? For instance, the cell has to move towards the right side. Now what is going to happen? That in the rear end, endocytosis will take place, which will result into the formation of endocytotic vesicle. This endocytotic vesicle will move to the other side where the movement has to, in the direction where the movement has to take place, and this endocytotic vesicle will fuse to the wall of the other side of the membrane, the direction in which the movement has to take place. Now, on one side, endocytosis is taking place, and on the other side, same vesicle is moving to the other side where this vesicle is being attached to the wall of the cell. Now, if you see, if endocytosis keeps on taking place from this side of the cell, after some time, there will be this wall of the cell will move forwards, or this wall will come around here, and this endocytotic vesicle, when it is attached to the opposite side, there will be continuous adhesion of the membrane on the opposite side, and this opposite side the membrane of this opposite side will move a little bit forwards. And the other point is, whenever this endocytotic vesicle is fused with the other side of the cell, there are also the receptors inside the vesicle. These receptors are being exocytosed, and these receptors will attach to the surface on which the movement is taking place, and the whole cell will get attached to these receptors just like this. And when they are attached here, the actin and myosin inside the cell will pull the whole cell towards the direction of the movement. So this whole, okay, now whenever there is movement, why the movement started in first point? This movement started because of some chemotactic substance. Now what is chemotaxis? Chemotaxis is that a substance is released, a chemical substance is released, which is attracting the WBC towards it. Now the movement because of this chemical substance is known as chemotaxis. Now there is a chemotactic substance here, and WBC has to move from this point to the right side. First step is absorption of membrane in the rear portion of the cell membrane. Absorption of the membrane in the rear portion of the cell membrane. And how this absorption is taking place? By the formation of endocytotic vesicles. Now, what will happen? These endocytotic vesicle will move to the opposite side, and a new cell membrane formation will take place at the leading edge of pseudopodium. Now, membrane is being absorbed from this side, and membrane is being added, attached, or sort of a new cell membrane is formed at this side. So there will be reduction in cell membrane on this side, and then there will be addition of cell membrane on the opposite side on the side of the direction of the movement. So new cell membrane formation at the leading edge of the pseudopodium by exocytotic vesicle formation. Okay, now this also is accompanied by the receptor proteins which are moving out. The receptor proteins are putated out. And when the receptor proteins move out, these receptors are attached to the surrounding tissues just like this. Now these receptors are attached to the surrounding tissues, and then the whole cell is attached to these receptors, and receptors are attached to the surrounding tissues. The whole cell is pulled in the direction of pseudopodium. Whole cell will be pulled in this direction towards the site of attachment by the action of actin and myosin present inside the cell. So this whole movement is known as amoeboid movement, and WBCs, fibroblasts, and germinal skin cells, they all perform amoeboid movement. Okay, so second one is the ciliary movement formed by cilia. So what exactly is cilia? Let's discuss as briefly its structure. Now this is a cilia, this is its tip, this is its membrane, and inside the cilia are the filaments, and down there is the basal body from where the cilia is being originating. Okay, now if we see the cross section of the cilia, the structure is like this. There are 9 double tubules, 1 2 3 4 5 6 7 8 and 9 double tubules and 2 single tubules in the center. 9 double tubules in the periphery and 2 single tubules in the center. And then in between these 9 double tubules, there are these red colored, these are dynein arms or dynein proteins. Now what is the function of these red colored dynein arms? The function is enzymatic. Now what exactly they do? Their function is they have ATPase function. They break down ATP and provide energy. Okay, so where exactly these cilia are found in the body? So there are 2 places where cilia are present. Number one is the respiratory airways, and the second one is the uterine tubes. Now in respiratory airways, the function of cilia is the cilia move like web. It has web-like motion as shown in this diagram. So cilia has web-like motion. So in respiratory airways, whenever the cilia move, they push the mucus over it towards the pharynx. The mucus is present in the respiratory airways. The function of the cilia is that by their web-like motion, they push and move this mucus over them towards the pharynx. Whereas the cilia present in the uterine tubes, the function of the cilia in uterine tubes is that they push the egg in the uterine tubes, in the fallopian tubes, towards the uterus. Okay, so the cilia in the respiratory tract and the uterine tubes, they have web-like motion. And other than the cilia, flagella also has ciliary movement. So flagella, where flagellum is present in the body? Flagellum is present in the sperms. So sperm has flagellum. Now there is difference in the movement of flagellum and cilia. Cilia follow a web-like motion, whereas flagellum follow quasi-sinusoidal wave type of motion. Okay, so this is the main difference. Okay, now let's get back in detail to the structure of the cilia. Now what exactly is axoneme? Axoneme is the combination of 9 double tubules, these ones, and 2 single tubules, these ones, and along with the protein dynein, which is ATPase activity, this one, the red one, between the 9 double tubules. So this whole structure, 9 double tubules with the single tubules with the dynein, this all is known as axoneme. Okay, so one is axoneme and the other one is the basal body. So this is the axoneme and this one label here is the basal body of the cilia. Now how exactly ciliary movement is going to take place? So what is the mechanism of its movement? So it starts with the electrochemical signal which is generated by 2 central tubules. The 2 central tubules will generate an electrochemical signal. Now this electrochemical signal will result into crawling of front tubules over the back tubules. Now, for instance, if the movement is going to take place in this direction, so these are the front tubules and these are the back tubules. Now, these front tubules will crawl over the back tubules. For instance, these front will crawl over these back tubules, and when they will crawl over them, there will be banding of this whole cilia as they are attached by dynein arms, the dynein protein, which is ATPase activity. So when these front tubules will crawl over these back tubules, there will be banding of this cilia backwards. So after they are bent backwards, there will be a sudden fast forward stroke, just like a whip. Now, if you focus on the motion of the whip, it first moves backwards and then suddenly, with a fast forward movement, with the fast forward stroke, the whip moves forward. So that is known as the whip-like motion. So this is exactly what is happening in the cilia. So where the energy is coming from for these front tubules to crawl over these back tubules, this energy is provided by the dynein arms, because this dynein arms has ATPase activity. So they will break down the dynein proteins, then acting enzymatically will break down the ATP and will provide the energy to the front tubules to crawl over the back tubules, which will cause the banding, and then there will be a sudden fast forward stroke, just like a whip. So this is amoeboid movement and the ciliary movement. See you in next chapter.